Hi and welcome to the PRIMA Lab Procedures overview video. My name is Sophie Weston and I'm the Clinical Trial Monitor for PRIMA at Menzies in Darwin. The first thing I want to reiterate is that safety is both a main priority for the participants in the trial and your own safety when working in the lab. Remember that blood products can be harmful and always use PPE when dealing with human samples. That may include gloves, sharp containers, and eye protection. So the protocol provides a detailed description of the required procedures for the trial. Along the top of the table here, you can see the time point after enrolment. And this includes every day that the participant will be attending the clinic. So the first column has day 0, the last column has day 63, and finally day of recurrence. And down the left-hand side are all the procedures that are required on that particular time point. What we're interested in today is looking at the lab procedures. So these include what procedures to complete after venous or capillary blood collection. The protocol table showed that there is a dried blood spot, cell-free HB, biomass, drug levels, serology, parasite PCR and host PCR analyses that will be conducted. And this table here shows the days and the amount and the aliquots that are required for those samples. The source is on the far left-hand side and on the far right-hand side, and it shows that whole blood will be used for the dried blood spot on all days that blood is collected. Plasma will be used for cell-free HB, biomass, drug levels, and serology. And the drug levels, this is a specific extra consented procedure on day 5 and day 6. Day 5 in Ethiopia only, and only in the intervention arm. And finally, the red blood cell pellet will be collected and frozen and used for parasite PCR as well as host PCR. So to make things easier for you in the field, we have developed a workflow diagram for all the procedures. It's color-coded with the days on the left, the procedure in the red colors, and the extra comments in the grey, and then where to store it. So these will be supplied to you at your field site. It can be printed, pasted on the wall, laminated. It's always available. If you do not have a copy, please get in touch with us and we will supply you with one. For the blood smear preparation, whenever possible, use separate slides for thick and thin films. Use reverse pipetting to transfer blood. Dry the slide, fix only the thin film with methanol, and the thick film should not be fixed, and protect unstained blood smears from excessive heat, moisture, and insects by storing in a covered box. So when the thick and thin films are completely dry, you can stain them. For screening, follow standard staining procedures. After enrolment, prepare one study slide. Follow the same procedures for every visit. Cover the smear with 10% V/V Giemsa for 45 to 60 minutes. Rinse the slide by flooding tap water, drinking water, or buffered water, and then dry the slide. So an overview of what the samples will be used for. The venous samples taken on day 0 and recurrence will be used for a microscopy slide, an SD biosensor which will give a G6PD reading and a hemoglobin reading, dried blood spot, and centrifugation at top speed for 10 minutes. And on the right hand side, you can see a small diagram of a blood tube, and this has been centrifuged. So you can see that the plasma rests on the top, which will be used for cell-free HB, biomass, drug levels, and serology. The buffy coat, and then the red cells pellets at the bottom, which will be used for parasite genotyping and host genotyping. If you are unfamiliar with cell-free HB, I'll give a short overview here, but feel free to ask any more questions should you require it. So the cell-free HB will be taken on day 0 and at recurrence, and it needs to be centrifuged 2 times. So to begin, centrifuge the vacutainer at top speed for 5 minutes. Aliquot the top 150 microliter of plasma into an Eppendorf tube, and then spin or centrifuge the Eppendorf at top speed for 5 minutes, and then dispense the top 100 microliters into a cryovial and store. Capillary samples are every day unless venous blood is collected. It's a maximum of 450 microliters in an EDTA microtainer, such as pictured here on the right. The capillary samples will be used for microscopy slide, SD biosensor for the G6PD reading and hemoglobin, dried blood spot, and they need to be centrifuged at top speed for 10 minutes. So with the capillary samples, we will take drug levels on day 5 if in Ethiopia, and day 6 for all sites.